So thank you very much for the invitation. I am very pleased and honored to present today actually one part of my PhD studies. As you said, I recently graduated in 2023, and my main research area is lipidomics, especially untargeted lipidomics, mostly applied into imaging mass spectrometry, which I will be discussing today with you, like small terms of imaging and maybe a little bit of applications based on the data that I obtained during my studies in my PhD. So the outline of today's presentation will be mostly about conventional lipidomics, which I believe most of you are familiar with, how we perform mass spectrometry imaging lipidomics, and mostly I will be focusing on the limitations that this technique has for us. So we all know the differences that we have between targeted, semi-targeted, and untargeted lipidomics, but most of all, these approaches have something in common. And the thing that they have in common is that through them we can identify substantial variances among experimental groups, and we aim to obtain the presence of a distinctive lipidomic signatures, either by specifically targeting it or by looking at it in a questionable way. For instance, if we perform untargeted lipidomics, at some point we don't really know what we are looking for, but we know that we want a lipidomic signature between the groups that we are comparing. And the question that I asked to myself during my studies was, what if we could visualize lipids in situ, like in their tissue or cell, revealing their special localization? And that can be done through imaging mass spectrometry, most specifically through MALDI, or matrix-assisted laser desorption. I want to put a little disclaimer here that I know that there are other ionization techniques in the world of imaging mass spectrometry. However, this talk today will be focused on the MALDI EMS. So in this presentation, in this slide, I will put the attention into the sample preparation, which is something very important when we perform imaging mass spectrometry. In this case, in my experimental design, I worked with human eyes. And in this part, you have a whole human eye globe in which I selected an important part of the human eye, which is the central part of the eye called macula, which is responsible for the central vision in humans. And in order to select this part of the tissue, I used a specific medical device which helped me to select the zone of interest. And then I performed cryosectioned slices of the tissue, of the interested tissue, about 14 micrometer in diameter, 8 millimeters. So the reason I did this was to obtain these slices and then apply a matrix which would help me ionize the molecules and prepare my sample, the tissues, for imaging mass spectrometry. So the way how we put the matrix onto these surfaces of the tissue, it's either by matrix sublimation, which is a device that we developed in our laboratory, or either by matrix sprayer deposition. Either way, if you would use the matrix sublimation, this is a picture that shows how the tissue would look. So the white matter, it's the presence of the matrix that has deposited into the surface of the tissue. We can say that it has a very homogeneous surface and distribution, which is very good for the signal that we will obtain after. After the section, it is prepared in such way, it will go into the mass spectrometer. And in this case, in my experimental design, we used a MALDI TOF, time of flight mass spectrometer, from Bruker. So here I want to discuss a little bit about the concept of MALDI. If we take, for instance, the tissue section that I mentioned earlier, here it's, for instance, the presence of the human retina, which is the wavy part responsible for our vision. If we would put a laser from the mass spectrometer, the laser will desorb the surface part of our tissue and it will mix it with the matrix. Thus, it will create some particle, like charged particles, positive or negative, which will then be detected by the detector of the mass spectrometer. So we can imagine something like that. So each part of the laser will distract one part of the tissue and we will obtain a specific chromatogram of the signal of the ion that we are looking for. For instance, if we want to have the intensity of phosphatidylethanolamin in blue, then we would have the ion signal distributed for this molecule in such way. If we want to look for another molecule, for instance, another phosphatidylethanolamin composed of 60, 0 and 80, 0, then we would have this kind of distribution. So something important to take from this slide is that two different molecules have different presentations or distribution in this tissue, and we can see this information only by imaging mass spectrometry. If we were to use a conventional lipidomics separation by an HPLC or something else, we wouldn't be able to see this lipid distribution in such way. So here I present to you a part of the distribution of another two ion signals. I'm sorry I cannot present to you more data, but this is part of some unpublished data. And we can see that here we have the human eye section, which is presented by the green part, which is the retina, once again responsible for the central vision loss, central vision perception. And here we have the distribution of two signals. So in violet, we have the signal of the phosphatidylethanolamine 18-0-22-6, and the other one in green, it's a phosphatidylcholine 18-0-22. So what is particular about this species is that they both have the same fatty acids in their positions, but they differ from their polar head. So even a slight difference in their structure, like the polar head, would make these lipids have a different spatial distribution in this tissue. So this is very important when we want to, for instance, compare control with disease groups, or only when we want to have a simple information about the distribution of these lipids in this kind of tissues. So most importantly, of course, that these techniques have lots of benefits for us, but I want to point out that these techniques have also lots of limitations. So the first one is the spatial resolution. So in this very nice publication, I found the comparison of different spatial resolutions of a specific biological samples, and we can see here, for instance, that when we increase our resolution, when we go up to 50 micron pixel, we can see that we have a high abundance of the ion yield. But when we decrease the resolution, the ion yield decreases as well, which means that we have to compromise, we have to put a compromise between the increasing the spatial resolution and decreasing the ion yield. It's very good to have a clear image. For instance, on the 0.6 micron pixel, we have an amazing clear image, which we can pair with our microscopic data and further and more. But we can see that the ion yield, it's barely visible, which means that we really need to increase a little bit more the resolution in order to have a higher ion yield. So this is like a daily challenge in imaging mass spectrometry, because either you have a very good spatial resolution, either you have a good ion yield. Nowadays, different manufacturers have developed or have put a second laser, or other people have modified the laser in order to perform or to have this good compromise between the spatial resolution and the sensitivity of your method. The second thing I want to put an attention is the effect of the matrix effect. So I said that the matrix, it's a chemical compound that you put on the surface of your tissue in order to ionize your tissue for better identification. It's important to know how you will depose this matrix. So I earlier mentioned that it's either with a sprayer on the first graphics, or either with sublimation. If you apply the matrix with a sprayer, you risk to delocalize the lipids. So the line, the unlimited line here, represents the confine between the tissue and the off tissue. So if you put the sprayer over your section and you don't desiccate your sample, then you will have the lipid signal off your tissue, and it's quite very significant high. It's about 200 micron pixel intensity, which means that you will not have your lipid ion signal there where the actual tissue it is. So you will have a derivation. But if you perform sublimation, we can see that we limit this error a little bit. And the perfect condition would be to perform the sublimation and to desiccate your tissue. So desiccation is important because it will evaporate, or it will take off all kinds of solvent tracing that are present. And we can see that if we do the sublimation and we put the desiccation step, we can see that our lipid signal will stay within the tissue, and we are sure that the lipid signal that we will obtain, it will be on the surface of the tissue, and we are sure that it will be localized there where it really is. For instance, here on your right, we have an example of my data. Here, once again, it is the representation of the human retina, and we can see that it's very subtle, but we can see on your first figure that through sublimation, we obtain a much higher intensity of our lipid compared to spray, which is a little bit more sparse and less concentrated. So once again, the sublimation yields superior outcomes compared to spraying, while desiccation enhances results in both scenarios. And lastly, it's the sample preparation artifact. So when we prepare a tissue, it's important to know if we are going to fix the tissue or not. So what is this fixing the tissue? It's using specific fixators like formaldehyde, which is a step that it's very common use in microscopy or histology, but it counteracts when we use this data or these preparations in mass spectrometry, because using fixators such as formaldehyde, for instance, it will or it might produce degradation products. For instance, I present to you here an example of from a very good article in which they seen that using a fixator such as formaldehyde will produce degradation products from phospholipids, specifically PE. Why? Because the amine part of the phospholipids will counteract with the formaldehyde, and we will have like methylated or demethylated phosphatidyl ethanolamines and a degradation of the inactive phospholipid. Also, hydrolysis has been seen because of the low abundance of the phospholipids and the high abundance of the free fatty acids. Therefore, it's important to know if we are doing the fixation or not. Sometimes you don't have the choice. You really have to fix your tissue because you are doing your analysis days or months after, and you are risking to degrade your tissue. Sometimes you don't. But it's important to take into account that several lipids could be degraded due to this approach. And with that, with the limitation, I conclude my presentation. I want to thank the collaborators that collaborated with the analysis of specific, especially MALDI analysis in different laboratories in France. I am very, very in awe with their help. So without their help, this presentation would be able, and I'm sorry that I did not give more examples of the different or other classes lipids, but due to the fact that this is an article in publishing, I cannot give more information about that. So I hope that that's all, and I am just open for questions. Thank you.
Thanks, Glenda, for the presentation. So we're gonna see if we have questions. Yeah, Justine has a question.
Yeah, thanks a lot, Glenda, for this very short but great presentation. I was wondering if, without saying your secret or all, but did you see the same species by liquid chromatography analysis of your retina versus imaging? Or
Yeah.
Yeah, it was quite the same. And did you notice in the MALDI imaging some big abundance of DG, which could be some, broken TG or things like that with fragmentation?
I understand. Yeah. So in simple HPLC mass spectrometry analysis, we saw a slightly degradation process. We saw a high abundance of diacylglycerols (DG). When it came to MALDI, the fact that we had only a few samples, like 2 human eyes, statistically speaking, I cannot make a significance that say they were important, but the abundance of the TGs was slightly higher than those of the TG. So probably there is a degradation process due to the fact that maybe the sample was not analyzed quickly after it was treated or I don't know, other experimental steps. This answers your first question. And when it comes to ratios of different lipid classes, such as phospholipids, cholesterol esters, and several other lipid classes, I basically the ratios were the same as those as I found on the HPLC and those that we found represented in the multi. Yes, it was basically that what we were expecting in terms of ratios.
Okay, thanks.
Next, we have two questions in chat. The first one is from Thomas. And how did you formally identify PE 18.18022.6 and PC 18.022.6 through MALDI? Did you use basically ion mobility or MSMS?
So I had three different approaches. So the second approach was to perform MS2 identification through Solarix FTR, so through the Fourier transform. So based on this MS2 identification, we were able to identify the structure based on the presence of the fatty acids. So that's how we identified the molecule and other molecules.
Thanks. And one last question in the chat, pretty long one. Could I ask in regards to delocalization with without desiccation, is this putting the matrix sprayed slide in a vacuum desiccator? Wouldn't the matrix tissue dry eventually anyway when put into the source of the Rapiflex?
So this is a very nice question because we actually had this happening. So when we used the sublimation, when we put the tissue into the module of the mass spectrometer, so the matrix was evaporated in an oval shape. So it was evaporated in the extremities of the tissue, but remained over the tissue. And this was because of a vacuum that was very high. But then we rearranged the parameters, and they did not happen. So for at least like 2 years of experimenting, I never had an issue after. So either with spraying or either with sublimation, the matrix would stay in the desiccator even for like 3 to 4 months. Of course, it wouldn't be very qualitative, but the matrix won't go away.
Thanks. So now it's time to move on to the last presentation of the day.